Well, we moved into this part of genetics, which we call cytology, the study of chromosomes. In the first part of these lectures, we talked about some of the basic terminology of cytology, cytogenetics, the study of both the number and structure of chromosomes, which is what we're really doing. Introduce some other terminology, such as karyotype, ideogram, and then we worked through some somes and ideas of what missing one chromosome was versus missing an entire set. And then we also talked about some potential ways that you might have some of these chromosome anomalies, such as replication events, multiple fertilization events, meiotic, mitotic, non-disjunction failures. What we want to start now is a series of lectures where we kind of look at some of the classic types of chromosomal anomalies and see how both those can be productive as well as be problematic, depending on which kind of system you're dealing with. Right? Now, you may remember that we divided up the chromosomal studies into changes in chromosome numbers, as well as then we're talking about changes in chromosome structure. So right now we're going to talk exclusively about changes in chromosome number. We'll start out talking about euploidy. Remember, euploidy is entire sets of chromosomes, haploid sets being gained or lost. Then we'll move to aneuploidy. Okay. Now, with euploidy, we really can't use good human models because there really aren't. It's not well tolerated at all by humans. I'm not aware of any living humans that are from an unfertilized egg or that are triploid or whatever. Now, there is mosaicism. Mosaicism is where certain body parts, certain body tissues may be triploid or something else. These occur during fetal development, and we'll actually talk some later about how this is a fairly common event, although mosaicism itself is not particularly common. We don't tend to see those cells actually make it to adult individuals. But so we're not talking about mosaicism. We're talking about entire systems where you have either reduced the number of chromosomes from a 2N component or you've added entire sets of chromosomes. Now, there are two forms that we'll talk about here. We'll talk about what is known as autopolyploidy. So remember what the ploidy means, that we're in euploidy systems. We're duplicating the entire genome. Poly means many, and just a general term for many. So now we're introducing the idea of auto. Auto in this case means self. So it's taking the same genome and duplicating it. As if you took your hand skin cells and rubbed them together and you took two 2N cells and made a 4N. That would be an autopolyploid event. The converse of this is an aloe polyploid. Aloe means different, just like allele meant different, right? Different forms. This is duplication resulting from two different genomes, usually at the species level or higher. And we'll talk about where those are meant, okay? So let's first look at autopolyploid. Very important. It's very important in the world of commercial plants. And many of the products that we actually use from commercial plants now after domestication are in fact polyploids of various forms, either polyploids by accident, we found them in nature, or they're polyploids by humans manipulating them so that they are. What you tend to see is that you see larger cell size when you have polyploids. So if you want bigger fruit, or you want bigger cells, to grow faster, you can do this by making them polyploid. And very often, because when you get them, particularly in imbalanced numbers like tetraploids or octoploids, well, imbalanced, but you end up having fewer or no seeds. So many seedless varieties, or things where you have fewer seeds, tend to be ones that we are production of polyploids. And I'll just give you some examples so that you kind of have this in your mind. Some triploids that are fairly famous: wine sap apples, which has very reduced seeds in them, of course, are classic triploids. Commercial bananas, many of the bananas, particularly those that are imported into this country, are in fact triploids. And so, these types of bananas are called Cavendish or Robusta. They tend to take a long time to actually ripen, even after they're picked, which makes them nice for transport. They also are relatively tough, so they don't bruise too easily. If you've had the pleasure of going into Mexico, Central, South America, you'll see there's several varietals of bananas that are available, including these very small ones here, which you can sometimes talk a graduate student into looking funny with. Very small bananas, very, very tasty. You can just eat an entire raceme at a time, very, very good. There are different varietals of these things. And if you go in different parts of the world, you'll see lots of different varieties. And it turns out that the Cavendish type, the export bananas, the ones that we receive, are actually a triploid. They're a triploid of a classic naturally occurring form, a musca, that we'll call type A here, and they're a triploid of that type A. There's other type A triploids that have different genomic components, which are the red bananas. These are dessert bananas. They're large, but they make great sugar content. So for flambés and things, they're used for that. They're highly prized in commercial kitchens for their ability. And then there's some that are not autopolyploids, but in fact we will call aloe polyploids because they are combinations of two completely different genomes. They're both muscas, same genus, but Echinata and Bellisona are the two forms that you see there. There's a variety, and multiple varieties of bananas. These aren't the only ones, but there's cooking variety that are based up are triploids, but they're two copies of the A genotype and one copy of the B, and they're high in sugar content and they're useful for cooking. And then the ones that we would call plantains in this country, they're very commonly seen here, are for the most part, and there are multiple types of plantains, but for the most part, they're one copy of genome A and two copies of genome B. And these are the plantains, the ones that you see, they're fairly large bananas in grocery stores that don't ripen. They're cooking bananas. They're commonly used and they're a staple. They're sort of the potato of the Caribbean, if you will. If you spend any time in the Caribbean, particularly Puerto Rico, you'll get tostones, which are cooked usually fried up a little bit and then mashed and deep fried with lime and things. Delicious. The other way is you just simply take them and open them up and cook them like a baking potato, and they will have very sweet form. But all of these are, of course, seedless, right? You don't get banana seeds, but in the natural populations, the fruit is really a seed house. So these are forms that have arrived from those. Right? Now, besides commercial bananas, some triploids, some varieties of seedless watermelons, which have these greatly reduced seed components, are there. And then there are vertebrates that actually, and they found primarily in the fish. Auburn is famous for some triploid catfish. These are produced by, at least in some ways, by taking females, uh, sort of putting in a pressure system, causing spindle fiber disformation, and they produce a 2N egg. You fertilize them with a normal male, and you end up with a triploid catfish. And these grow off very quickly, so that they're not in the pond as long, and so they're very adaptable for getting food to a usable, presentable size quickly. There are trout that are raised in hatcheries that are triploids that produce these massively large trout that have been released and some people like to catch. And then there's other useful ones such as grass carps have been produced that are triploid sterile grass carps so that you can release them. They're non-native fishes, but they eat this invasive vegetation and clean up a lot of the waterways where this vegetation is taken over. And if you go to South Carolina, North Carolina, Georgia, and some places, you'll actually see where they've been released to control vegetation. Then there's some tetraploids. There are a number of these. I think probably the one group that you're most familiar with might be potatoes, and this is a sort of a collage of different potato types. There are many, many potato types, and some of them are, in fact, autopolyploids, autotetraploids, and they are found now. Potatoes are wide-ranging. Now they were originally from South America and the Andes, even if you go into the Andes, you'll find that there are some normal diploid ones. There are some triploids, and all the way up to pentaploids, etc. But many of the common varietals that are used throughout the world are actually a 4N autopolyploid, so they've duplicated. Now these have to be reproduced. They don't produce seeds, so they clonally are reproduced. So if you're going to raise potatoes, what they would do is just cut an eye out and plant the eye of the potato, which grows in these spouts, and then you get new potatoes. And of course, that was the main issue. This is Ireland. This is Western Ireland here in Connemara. And if you go there, even today, you'll see the scars on the side of the hills from these potato plantings. back in the early 1800s. There was the primary food source for the Irish people, certainly those outside beyond the Pale, living in the western parts of Ireland. Pounds of potatoes were eaten per day by families, and then of course had the couple of series of what are known as potato famines, where disease came in, hit the potato crops, and just decimated the crops across Ireland, and causing people to migrate. In fact, some of your family may be in this country because you were part of that migration that occurred. Some of it was forced migration, unfortunately, due to religious and political systems. Others was just simply because of lack of food and availability. But you can still see these scars that are found there. The reason that the famine was so bad is that these potatoes were being produced clonally by cutting that. So it meant that essentially every potato in Ireland was the same stock. So once a disease could hit one area, they could just easily spread across the other. There was no natural resistance because they were all essentially the same, one big potato, if you want to think of it that way. Okay. Other things that we can see is there's some octoploids that are very important for us, such as commercial strawberries. Those big, beautiful strawberries that we use for daiquiris or smoothies or things. Wild strawberries are much smaller, and they have lots of seeds on them, which are not, found. And of course, commercial strawberries are propagated by cuttings and tissues and other things. There's some other forms of this. The naturally occurring one, good friend of mine works in northern Michigan, and his research projects are on Great Lakes sturgeon, these large sturgeon, trying to recover those. They come up into the streams and rivers in this time, the spring of the year, late spring, and spawn. These are octoploids, fishes. Makes them very difficult to do genetics work on because they got so many copies of every chromosome and every gene, but they're very interesting animals to work with. And in fact, just to kind of introduce another term, there's what's known as polyploidy. Paleo means old or going back in time. Throughout lots of lineages, including the vertebrate lineage, we see these duplication events where the genome, 2N genome, gets duplicated to a 4N genome. And that has certainly happened in the teleost. The lower teleost, and many of those are what would be 2N. There was an event then that occurred in the middle teleost, which produced 4N. Later on in amphibian times, there was another copy that duplicated all those, which means that you go from having 2 copies of a gene, to now having 4 copies of that gene, and allowed these copies to multiple things happen. Sometimes the gene copies have just become non-functional. We call it pseudogenes. Other times this copy that's out here can be modified to maybe work differently in the eye or in muscle or whatever. And we see these whole changes, these forms that occurred. But we know that these occurred, and they occurred in lots of lineages. It's called polyploidy. Now, it's not only important for major groups and to expand, but also in individual plants, we would see it when we talk about some of those. Okay. All right. So just to kind of work through, as your book does, through the process of polyploidy, looking at how autopolyploidy might occur through mitosis and meiosis. This form right here is in mitosis. Now, again, I really dislike the way this is set up because it implies that replication is part of mitosis, which we know it isn't, right? So, even your textbook and many textbooks are just really lax in how they represent this. Anyway, for us, replication has occurred. We get to mitosis, and we simply get a non-disjunction, a complete maybe non-disjunction, which ends up giving an autotetraploid cell. Now, these cells, of course, are not going to be part of the gametic system, so they're going to only be in that individual, but they do occur, and you get autotetraploids that can occur from that. If it's a plant, this might become a plant then that's an autotetraploid that could reproduce clonally or in some other way. Okay? Now, through meiosis, it can occur in multiple things, but again, non-disjunction can occur. You can then get fertilization and end up with triploids or all sorts of forms by having this non-disjunction that occurs here. Okay, and you can have it, of course, in other ways. You can have just dual replication and all those events that might occur. This is how your book explains it, and I think they're pretty good examples, except for the fact that replication seems to be involved in meiosis here. Okay, now the other form of all this is what's known as aloe polyploid. All right, so we're going to put different genomes together. So I'll give you a few examples of that. One that's very important throughout this country is cultivated American cotton. People wondered where cultivated American cotton came from, these sort of tight balls, midland leveled fibers, so they were easily mechanically separated, which allowed the system to develop, where we had cotton gins and things because we could pull the seeds out fairly efficiently. Well, these appeared in North America and became American cotton. Old world cotton and wild cotton sort of looks more like this. It's long fibers, kind of spread apart, more difficult to manage. When people started looking at this New World cotton, this American cotton, they realized it had 23 pairs of chromosomes, 13 large and 13 small. There was actually some work done at Tuskegee where they took and basically took some old world cotton, some weed-like cotton, a cotton plant that was a weedy species from North America, fused them together, and they actually ended up with cotton that looked very much like American cotton. But more recent evidence suggests that this form of cotton has been around a lot longer than just, since Europeans came to North America and brought old world cotton with them. And in fact, it represents a very long period back from an introduction, some other type of introduction. And cotton itself can be traced back 5 to 10 million years ago, and it split at that point into what are the New World cottons and groups of cottons. Okay, these have a genome size of about 885 million base pairs. African cottons, the old world cottons, were much larger, almost were twice as large as far as their genome size, and they developed into all sorts of different varietals that ended up in not only in Africa but all the way into Australia as basic forms of cotton plants. Again, not necessarily the harvest plant that you think of for using for fabrics, but plants that represent are in the cotton family. What is now considered American cotton, this aloe polyploid, arose from a combination of some of these African varieties and one of the New World varieties, but it occurred somewhere as much as 2 million years ago that the initial hybridization and to produce the aloe polyploids occurred. And we can see that it's a large genome kind of mix of these two, and it diversified into 5 species from over those couple of million years in the New World, became New World cotton. So it wasn't a recent event. It turns out to be one that goes back at least 1 million years, maybe further, that we've had this sort of form of cotton around. But in terms, it's still an aloe polyploid. Now, just the book goes through, and I wanted to highlight some ways that you might produce these. This is an example where you have gametogenesis. You fuse those together, produce some type of hybrid, which goes through, because of disruption of that system, maybe go through non-disjunction or just skip over the mitotic cell division. And then you get an aloe polyploid. Okay, and that aloe polyploid then, if it's got a sort of a good combination of gametes, is actually able to produce functional gametes that will combine with itself to work. One system that's somewhat evidence like this is the wheat varietals that we use to make flour for cooking. Wheat is known to have to be in the result of multiple paleo hybridization events, where you took wild grass and an elkhorn wheat, those fused together, then you ended up with one of these non-disjunction events to produce a different type of wheat that then combined once again with a different form of wild grass to produce a hybrid. So some mitotic non-disjunction, you ended up with some of our varietals of what we would think of as bread wheat that we use today. Some of the forms, these are 6N, so forms, and they can be very easily, they can be traced back to all of these events that occurred over time. Okay, another form of aloe polyploid are commercial cultivated sugarcane, which are grasses, forms that we extract much of our cane sugar from. They're found all over the world, but different varietals of it, but is an aloe polyploid that can be used. And peanuts, very important in the southern United States after the loss of the cotton crops and things due to boll weevils. Many people converted over into sandy soils to growing peanuts. Commercial peanuts are an aloe tetraploid. They are AABB, meaning they came from 2 naturally occurring varietals that were present, aderensis and others, and these are formed up together to produce this AABB, aloe tetraploid. 2 copies of one, 2 copies of the other to give us what we think of as Spanish peanuts or commercial peanuts that are grown pretty much throughout the world. As I said, most of these polyploid type things are not particularly common in vertebrates or anything, but there is at least one case that's been argued that it represents an aloe polyploid event. The red viscossus mouse is found in South America, and it has, if you genotype it, it clearly has chromosome sets that represent a combination of different species. And they've been recently has been some argument about what those species are and whether they, if they contributed. But there does appear to be a nice little aloe tetraploid event, so where there's been a complete fusion of 2 different species of mice together to produce these. Now, what's really interesting about them, and I'll say up front that there's argument that that's not what's happened, that it's been a duplication event of a single genome, but some very, very recent work suggests no, they're going back the other direction, that there really are chromosomal sets that represent ancestral species that have fused together completely. Now, one thing's interesting is they have, in a females at least, they have 4 X chromosomes. They have ligase mode, so they have XXX, Y in males or XXXXX in females. But it turns out that only 2 of these X's are actually functional. The other 2, if this were 2 X's, are actually become semi autosomes. They don't participate in sex determination or linkage or anything else. And one of the reasons they know that is they go look and look for bar bodies and particularly mark which ones of these chromosomes they're working with. They can see that there's only 1 bar body. If these were functioning as sex chromosomes, you'd expect 3 bar bodies to be present. But what's happening is one of the pairs is actually not functioning as sex chromosomes anymore. It's functioning as something else. So that's pretty interesting. Okay, and I want to end this talking about just an odd form, but an important form. This is called endopolyploidy. Now, endopolyploidy relates to that whole concept of mosaicism, but endopolyploidy is something that occurs fairly regularly, right? It's not really a chance event, in the sense that you can kind of go and look at certain body parts and predict that you'll find some polyploidy there. So this is where certain cell lines are polyploid in a normal 2N organism. Okay? So we're just talking about certain cell lines. And some examples would include vertebrate liver cells. Your liver is a unique organ. It's one of the few internal organs, or only internal organ, that can actually replicate itself, reproduce in mass size. So if you lose part of your liver, you can replace it. Some liver cells, or populations of liver cells, are 4N. There's some populations that are all the way up to 8N, and some, a very few that can be found that are 16N. Now, the vast majority are 2N or diploid, but you have these components. People wonder why that might be. I don't know that there's a good explanation. Some theories have been that, well, the vertebrate liver is a detox center, and so we were primarily originally, for us at least, and for many vertebrates, we eat vegetation. Well, plants don't like to be eaten, so they put all these toxins into their leaves and other parts, and so our livers become centers for detoxing that. A lot of those toxins are actually mutagenic. So if they mutate some of the DNA that we have in a cell, if you're a 4N cell population, you've got other copies that you can then turn off the bad ones and use the good ones. Now, how cells recognize good and bad is for debate, but certainly DNA recognition of bad DNA goes on. So it's one idea that's why they're there. No one actually, to my knowledge, knows, but they certainly exist. Okay, just to give you a really, and there's Drosophila, there's the special lines, other things. One really cool example, and the reason this picture here is Geras, water striders. These are not spiders; they're little Geras. They're found commonly in very quiet areas. If you happen to live on a lake anywhere in the southern United States, just go back into very quiet water, and you'll see these little guys skimming across the top of the water. They're called water striders. Water striders in the larvae. The salivary glands of the larvae have this sort of runaway replication without mitosis. And some of the cells in there can be found to be 1024 up to 2048 in these salivary glands. Now, if we think about it, these guys have 22 chromosomes. So if you do all the math there, some of these cells may have as many as 40,000 chromosomes in them. I don't know what poor undergraduate or graduate student was originally tasked to count all these, but that's the current estimate. So they're just packed full of these polytene and chromosomes, which are obviously fill the cells up and make them essentially non-functional. So I mean, it's a runaway system. I don't think it's there for any evolutionary reason. It's just a system that, for whatever reason, is out of control. But again, it's only certain parts of these salivary glands and only certain cells, certain cell lines. It's endopolyploidy. But it's a fun thing if you sitting around, your parents or your significant others want to know if you've learned anything, you can say, hey, you're a polyploid, and point out the fact that their liver, because they're vertebrates, so humans and mammals, vertebrates, their liver cells, they're polyploid forms. Okay. All right. So we've worked through the big parts of euploidy. The next lecture series, we'll start talking about aneuploidies. And with aneuploidies, they certainly occur in humans, very commonly occur in part of these human systems. So we'll use mostly human examples when we talk about those.